Hello students, how are you? I hope you are all doing fine. I've already came to know that Dr. Zoha had discussed about the replication and transcription process. Let's talk about translation today. What is translation? So we all know chromosomes are nothing but long strands of DNA, right? So it is a strands of DNA I have drawn. And in the DNA, there are specific segments that we call gene. These genes can control or regulate specific trait of an organism. So how do they do that actually? They do this regulation by synthesizing specific sequence of polypeptide chains, or proteins, the functional proteins. These genes can actually exert their control in a specific trait by these specific proteins. So the thing is that the gene is there, which contains the genetic information of the base sequence of the proteins, and these genes first are transcribed into messenger RNA, and this messenger RNA conveys the message to the cytoplasm, ribosome, and produces a specific protein. So this whole process from gene to protein is called the central dogma of life, right? And this specific portion of central dogma where the messenger RNA is converted into protein is called translation. So it's a very important step in gene expression process. So what is the definition? Is the definition of a translation? You can say it is the synthesis of protein according to the base sequence of messenger RNA. So synthesis of protein according to the base sequence of messenger RNA. These are these two important words here. So what is the big picture? What is the main thing that happens in translation? Here you can see this is a messenger RNA. It's a strand of messenger RNA. And it had got the blockades of codons here. You know what is codon? Codon is triplets of base sequence, which can actually refer to or indicate specific amino acids. Like AUG, this codes for methionine. So whenever there is AUG in the messenger RNA, a tRNA will be attached, which has got the methionine in it. So specific tRNA with specific amino acid will be attached to the specific codons. Just like that in here, there is another amino acid according to that matches with the code. There is another amino acid that matches with the code. So this goes on. Whenever there is a stop codon, this production of amino acid stops. And meanwhile, all these amino acids are stitched together by the peptide bond, and ultimately the polypeptide chain is formed. And that's the basic idea, the principle of translation, the big picture of translation process. So what are the requirements of translation process? First thing first, you need a messenger RNA, mRNA, which contains an initiating codon that actually initiates or starts the translation process. And there must be a termination codon where the translation process will be terminated whenever it is required or whenever the proper code is there. We need a tRNA. Of course, we need a tRNA. These are different arms of tRNA. So tRNA has got amino acid acceptor arm, and there is an anticodon arm where the anticodons are there. So these anticodons actually search for the codons in messenger RNA and stick to the codon that it matches, right? So we need a tRNA. We also need specific amino acids, which is attached to the tRNA. We also need protein factors, which initiates, elongates, and release the amino acids. We also need the ribosome. Ribosomes are actually the platform of protein synthesis. So we obviously need a ribosome. We also need ATPs and GTPs, and they are required for the synthesis of the peptide bond. So another thing, we need amino acid tRNA synthase is another enzyme which also called the tRNA ligase, and it helps to ligate the tRNA with the amino acid. So these are the requirements of the translation process. Now let's delve into the actual process of translation in details. So first thing first, we will need to initiate the translation process. So we need a pre-initiation complex. And how is it formed? The pre-initiation complex has got met tRNA. That means the tRNA which has got the methionine as an amino acid. And there is 40S ribosome. It is a small subunit of ribosome, which is attached with the met tRNA. And there is initiating factor and also a GTP. So in this picture, let's see, as you can see, this is a pre-initiating complex. This is a pre-initiating complex, right? Here we got methionine. Here we got tRNA that contains the methionine, and also got the smaller subunit, which is 40S ribosome. All these things make up the pre-initiating complex, and this pre-initiating complex, whenever a messenger RNA is presented to it, it clings to the 5' end, and it moves towards the 3' end and search for the initiating codon, that is AUG. So it first attached to the 5' end of the messenger RNA, and it moved towards the 3' end, this 3' end. And whenever it is positioned against the AUG, that means the initiating codon, it clings to it. It stops there. So after the attachment, in this picture, you can see the pre-initiating complex moving towards the 3' end. Now, just the moment it is positioned against the AUG, another subunit of ribosome gets attached, and it is called the 60S ribosome. So the next step is the synthesis of ATS initiation complex. And this ATS initiation complex contains the previously made pre-initiating complex. It contains messenger RNA, and it also contains the newly arrived 60S ribosome, which is a bigger subunit of the two. And also in this 60S ribosome, there are 3 sites: P site, that means peptidyl tRNA binding site, the amino acid tRNA binding site, and the exit site. So in this picture, you can see this is the 60S ribosome, the bigger subunit, and it had got the peptidyl tRNA, where the peptide bond actually forms. And the A site, that means the proper amino acid containing tRNA, it will get attached to this site step by step. And this is the exit site, where whenever the peptidyl tRNA moves to this site, it is exited from this portion. So the whole thing actually moves from 5' to 3' end, from 5' to 3' end. So the sites will be shifted from here to here, here to here, right? So in this picture, you can see in the P side, the initiating codon AUG is there. So whenever it is attached, the met tRNA is attached in the P side, another tRNA containing the proper amino acid will be attached to this codon 1 side. So not any tRNA will be attached here. Only the tRNA will that will be meshed with this codon in this side can be attached in the A side. So this is the way the tRNA is attached. The new tRNA is attached to the A side. So now in this picture, you can see whenever the new tRNA or amino acid tRNA is attached, the amino acid in the P side actually links with the amino acid of the A side. So there is a formation of peptide bond here. Next picture you can see this bond is already formed. So this amino acid from this side jumps into this side, from P side to A side, and these things goes on. And then on the next slide, you can see the total 60s total initiating complex actually moves toward the 3' end, and the tRNA that was in the P side earlier is now in the E side, that means exit side. So this residue of the tRNA will be exited from the E side. And another new tRNA with specific amino acid will be attached to this next A side. So this goes on, and this amino acid jumps into this place and will form the longer polypeptide chain. And at some point, when there is a stop codon here, then there will be the termination of the protein synthesis, right? So there's the thing. So it is easier to actually remember seeing the pictures, not by the descriptions. So if we can see the whole picture serially, one by one, this is when the pre-initiating complex was formed. That means the 60S ribosome, 40S ribosome, along with the tRNA. So when it finds a messenger RNA, it clings to it. And after that, it keeps scanning, right, from 5' to 3'. And whenever it finds the AUG, it clings together, and the translation process starts. After that, the 60S ribosome comes into play. 60S ribosome got 3 sides: E side, P side, and A side, right? And after that, the tRNAs actually shift their position from A side to P side to E side, then exit the side. So this process actually keeps the amino acid from P side to S side. They keep jumping from P side to S side one by one after another, and ultimately this peptide chain produces longer polypeptide or proteins. So this is basically the process of translation. So after the translation process, you got all the amino acids and protein. This is not what you can say. This is complete or active proteins. These are not of any use unless or until you do some modifications, right? So there are some modification must be done for the protein chain or the polypeptide chain to be activated. So what are the modifications? There are some modifications. So these are called post-translational modifications. It is the chemical modification of protein synthesized through translation to make the protein functionally active. It is done by 6 different mechanisms. Number 1, removal of N-terminal methionine. Remember, methionine actually is required for the synthesis of all the proteins. So methionine starts everything. But after the completion of the translation, they must be removed. Otherwise, the protein will not be activated. So number 1, removal of methionine. Next one is limited proteolysis. That means the protein must be degraded in very specific areas. Otherwise, they cannot be converted. They cannot be activated. So This proteolysis, there is an example, for example, pepsinogen produces translation is modified by pepsin by limited proteolysis. So in our digestive system, we all know these proteolytic enzymes remains as inactive form. So these are actually done by limited proteolysis. This activation is done by limited proteolysis. Then hydroxylation, addition of hydroxyl group to a specific amino acid will lead to the activation. For example, lysine and proline of collagen are hydroxylated after the synthesis of collagen. Then gamma carboxylation, and it happens in the coagulation process. Then again, glycosylation and phosphorylation. These are other post-translational modifications. In fact, phosphorylation is one of the most common post-translational modifications. That's why I have actually marked it at red color. It's very important. Most actually common post-translational modification is phosphorylation. That means addition of phosphate group. And glycosylation means addition of carbohydrate group. So after the post-translational modification, all the proteins are activated. They can be activated, and they can exert their functional effect in our body. So that's it. That's all about the translation process and all the things that goes into it. And I hope you have got a good idea about the translation process. Now we will watch a video showing all the process of translation in a very concise way. Let's watch it.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.